I'd like to invite the next speaker, Jennifer Cohen. Jennifer is from the Sundaram lab in UPenn, and she'll tell us about a transient pre-cuticular apical extracellular matrix that defines tiny tube diameter. What tubes, I guess we'll have to wait and hear.
That's right, but only about 4 minutes. Oops, okay. Biological tubes transport liquids and gases throughout our bodies. And these tubes overall have the same general organization with an apical surface lining the lumen and a basal surface outside. But they come in dramatically different shapes and sizes. The very smallest tubes, such as those found in capillary beds, can be unicellular, meaning they're composed of only one cell with an intracellular lumen. And these tubes can be either seamed, meaning they have no junction, or meaning they have junction along their length, or seamless, meaning they have no junction along their length. Because these tubes are so tiny, and because they have this unusual junction structure, there might be some really interesting biology involved in allowing these tubes to achieve and maintain their shapes. I'm interested in what those are. And this is a really important question, really because loss or abnormality of capillaries is associated with stroke and hemorrhage, and constricted capillaries is linked with heart disease. But although capillaries are clearly very important for human health, there's not a whole lot known about how these tiny tubes get and keep their shape, in part because it's just very difficult to visualize these small tubes in mammalian systems. But fortunately, C. elegans offers a genetically tractable and easy to visualize model in its excretory system, which is composed of these 3 tandem unicellular tubes. And this system is responsible for the animal's osmoregulation, which is thought to occur when the canal cell, shown here in red, takes up fluid from the animal's body cavity and then passes that fluid into the duct cell, to the pore cell, and into the outside environment. The duct and canal cells are both seamless tubes, and in particular, the duct undergoes a pretty dramatic morphogenesis event from this simple shape that it has during embryogenesis to a much more complicated, elongated shape later on. I'm interested in what allows the duct cell to undergo this morphogenesis intact. So one thing that we think might be involved is apical extracellular matrix, or apical ECMs. Apical ECMs are secreted onto the apical surfaces of most epithelia, including the lumens of tubes such as capillaries, for example. And apical ECMs, we can see, this is a electron micrograph of a capillary, and it's a little hard to see on here, and I'm sorry, but there's multiple apical ECM layers here, and this is a zoom in along the membrane. And these layers are a pretty common feature of apical ECMs, which are typically composed of proteoglycans and glycosaminoglycans. The duct cell also has an apical ECM. Shown here, you can see this fibrous stuff in the middle of the lumen. Now this fibrous stuff later gets replaced by a collagenous cuticle that is going to line the lumen later on, after the duct cell undergoes its morphogenesis. We really don't know anything about what these components are that form this duct apical ECM, or what its function is prior to cuticle appearance. So one thing that we do know is in apical ECMs are zonopellucida domain proteins, or ZP proteins. These proteins were originally identified in the oocyte's extracellular coat, which is also called the zonopellucida. In this electron micrograph of the zonopellucida, you can see it's really big and thick, and ZP proteins are the main components of this extracellular matrix, where they assemble to form higher-order structures such as polymers or meshes, as diagrammed here. But outside of the zonopellucida, ZP proteins are highly abundant and present in many apical, including luminal matrices, such as those within the gut and kidney tubules. However, there are also their loss is associated with a long list of human diseases, notably vascular and kidney disease. And in model organisms, they are known to shape semiapical surfaces. But there's a lot that we don't know about how they form these matrices or how they're actually able to shape cells. So we did identify a ZP protein, LET653, that is a component of this duct pre-cuticular apical ECM. And the LET653 encodes several isoforms of a secreted protein that all have this basic domain structure shown here. With these two pan domains, also found in the blood clotting factor plasminogen, which might be protein-protein or protein-carbohydrate interaction domains in LET653. Next, LET653 has a ZP domain whose two halves are spanned by a protected mucin-like region. Mucins are also ECM components that are also highly glycosylated. So here's a LET653 fusion protein in the duct lumen during embryogenesis when the duct looks like this, and then slightly later when the duct is much more elongated. So we wanted to know when and where LET653 is expressed, and it turns out that it's specifically expressed in the duct and pore, but not the canal and the excretory system. And LET653 is present in this early pre-cuticular apical ECM. So it's present in the embryo, goes away, and then reappears before a new cuticle is synthesized. Now, we wanted to know why LET653 is in the duct. So we used an apical membrane marker to look at the duct cell in both wild type and LET653 mutants. And in wild type animals, the duct forms and there's the lumen, while in LET653 mutants, the duct lumen at this stage looks identical to wild type. However, slightly later in wild type, the duct lumen continues to elongate and to narrow somewhat, while in LET653 mutants, the duct lumen fragments and fluid starts to back up behind this block shown here. So to summarize, LET653 is in the lumen early on, before cuticle matures. And in LET653 mutants, while the duct initially forms normally, later on, as the duct lumen begins to both elongate and narrow, that duct lumen fragments and fluid eventually backs up behind this block. So based on these data, we hypothesized that LET653 might be required to expand the duct lumen to prevent over-narrowing and fragmentation during morphogenesis. So first, we wanted to ask what domains are important for LET653 function. So we generated these fusion proteins and found that the full-length LET653 and the LET653 ZP domain were both able to rescue duct lumen defects, while the pan domains were not. Now, we wanted to know whether LET653 was actually able to expand a lumen. So we expressed our LET653 fusion proteins in the C. elegans gut, which is a much larger multicellular lumen that does not normally express LET653 or develop a cuticle. And the full-length protein was able to expand this lumen, but the pan domains were not. So again, this is consistent with the idea that the LET653 ZP domain might be able to expand the duct lumen. So we thought that the LET653 ZP and pan domains' different activities might be due to these domains' interactions with different apical ECM layers. Because the duct is so small, we couldn't see these different layers, so we turned to the much larger multicellular volvo lumen, which is shown here in this electron micrograph. You can see multiple apical ECM layers here. There's a fibrous layer, zoomed in here, and then at least two layers along the membrane here and here. So we found that full-length LET653 in the vulva was able to localize both along the membrane and to that middle fibrous region, while the pan domains localized only to the fibrous region, and the ZP domain could localize only to a subset of the membrane, suggesting that the ZP domain is, or that the pan domain is able to localize the ZP domain to the membrane. Now, we know that ZP domains can form stable higher order structures in other systems. So we wanted to know whether LET653 was also forming such higher order structures along the ZP-bound apical membrane. So for that, we turned to fluorescence recovery after photobleaching. We photobleached LET653 both along the apical membrane and in the center that's bound by the ZP domain. We found that LET653 recovered very quickly in the center. You can see it here, and here it is quantified, while LET653 that was bound to the membrane recovered very poorly, indicating that LET653 is likely stably bound and incorporated into a matrix of this membrane. Now, to summarize, LET653 is able to expand lumens via ZP domain. LET653 is present at these two layers, here and along the membrane, and we propose that this might be true as well in the duct cell. LET653 is captured via pan-mediated interactions and then is able to incorporate stably into some sort of matrix structure via its ZP domain along the membrane. And this interaction is able to expand the lumen and might also direct where cuticle is later synthesized. So finally, I'd like to thank the entire Sundrum Lab, particularly Hasreet Gill, who was my co-first author on this project. And also I'd like to thank David Hall, who provided the electron micrographs that I showed you here today. Finally, this work was recently accepted into PLOS Genetics, so you'll be able to read it in there soon. All right, thank you.
So we have time for questions. I'll ask the first. So what's the difference between the glycocalyx and extracellular, well, this kind of apical extracellular matrix? Because it seems like they both have glycoproteins and they're bound to the membrane.
I think it's primarily semantic, but there might be someone else might have a different opinion.
And the red protein in your model that links between...
We're really interested in finding out what that is. We have some ideas. There were some other things that came out of our genetic screen that led us to LET653, but it's in the works.
Oh, I have one question. You have two ZP domain in this protein, and can owning the C-terminal ZP domain, just one ZP domain, can rescue your phenotype? I just want to know if they are redundant.
Right. The two halves of the ZP domain are not identical. We're actually testing that now. In other molecules, the N portion of the ZP domain is able to polymerize all by itself and actually might have its own function. Although the C-terminal portion of the ZP domain can have very specific binding partners, so we're testing that. Hi.
Did you ever notice any expression of 653 in the cuticle, especially when you made the deletion mutants? Did it float out there or partake in cuticular things?
We've never seen it inside of a structure that looks like cuticle. It's not expressed in adulthood, so after all of the molts are finished, it's gone. Where we do see it, however, is in between the worm and its cuticle that it's trying to shed. So we really think that it's a pre-cuticular thing, and it's maybe setting up that cuticle. But, yeah. Hi.
Have you screened other signaling molecule like TGF-beta to see if they function in the same pathway with this zona pellucida protein? Like maybe this one could be retaining some signaling molecule or releasing it.
Yeah, that's a great question. We haven't done that, although there's definitely models in the literature where that's been the case. Although here it does seem like there's likely a physical job for this protein to do as well. Thanks.
So following up on that, Jennifer, so your model says expands the lumen, but could it be that it's just mechanical support, like an internal cuticle while the external cuticle is being shed?
Yeah, I mean, it's definitely possible.
Okay, so if there are no further questions, let's thank Jennifer once more.